Chapter 3, the cellular level of organization. So we are working our way up the hierarchy of life. Last chapter, we studied about atoms, bonds, how they come together to form molecules. Now, molecules will form uh, cell organelles, and all of them come together to form cells. Some of the most important learning objectives in this chapter is we will understand what cell theory is, and also the basic anatomy and physiology of the cell, the different organelles, their structure and function. We'll talk about the plasma membrane or the cell membrane and some of the properties of the cell membrane. It's one of the most important organelle because it's important for maintaining homeostasis. We'll study diffusion, osmosis, and transport of molecules over the cell membrane. We'll briefly discuss the central dogma of biology, which talks about the flow of information in a cell, how the DNA in the nucleus transcribes to form a messenger RNA, and then that acts as a template for protein synthesis. In other words, the DNA dictates the amino acid sequence in a protein. We'll briefly touch upon the main events that happen in the cell, like mitosis, DNA replication or duplication, transcription, translation, and gene expression. Cell theory. So, what is the cell theory? It proposed by Hooke in the 16th century, and it says that all life is made up of cells. So, if you remember the 6 kingdoms of life, archaebacteria and eubacteria, they are prokaryotes, which are simple life forms, mostly unicellular. And then there's eukaryotes, which is protist, plants, animals, and fungi, which are usually multicellular. There's also unicellular protist. Prokaryotes, they don't have a membrane-bound nucleus, whereas eukaryotes will have a membrane-bound nucleus. So Robert Hooke proposed the cell theory. Cell theory says that all life on Earth is made up of cells, and all cells come from pre-existing cells. Cells are the smallest unit that can perform all the vital functions necessary for life. And every cell, in order to survive, will maintain homeostasis. So, in anatomy and physiology, when you talk about disease, it starts at a cell level first. That is why we need to know cells thoroughly. Our body is made up of like trillions of cells, and each cell has its own function. The cells will provide the structure for the body, and it will have nutrient uptake and getting rid of waste, and they'll carry out important functions. The scientific study of cells is called cytology. There are 2 types of cells in our body: the sex cells, which are haploid, which will have half the number of chromosomes, and the body cells or the somatic cells with 2 sets of chromosomes, 46 chromosomes, that is 23 pairs. After the discovery of advanced microbiology techniques for labeling the pictures of cells that we see in the books, they light up. It's beautiful. It's called fluorescent microscopy. So here, like when the sperm cell and the egg cell unite to form a zygote, how do the different types of cells in the body develop? It all comes from one cell, which is like a stem cell. They can differentiate to form muscle cell or fat cell or bone cell. This is called cell differentiation. So even though they have the same genome, a cell, because it is uh, totipotent or pluripotent, it can give rise to other cell forms. So this process is called differentiation. Cell differentiation is a process by which cells are becoming specialized to perform a certain function. For example, a muscle cell has to contract, so it has a very unique anatomy and physiology. So stem cells can either be totipotent, that means they can give rise to any other cell type, or pluripotent, that means a restricted number of cell types. And here's a flow chart that shows how the different blood cells in our body, they come from a totipotent embryonic cell in the bone marrow. The plasma membrane or the cell membrane, which is the outermost boundary of a cell, is considered to be one of the most important organelles in the cell. It is made up of a phospholipid bilayer. So we know lipids have 3 fatty acid chains. One of the fatty acid chain is replaced by a phosphate group. So it's called phospholipid. So this is a very unique molecule. It is called amphipathic. That means the phosphate group loves water, and the fatty acid tail is hydrophobic. So we have a hydrophilic end and a hydrophobic tail. So it can self-assemble to form phospholipids. So the heads that love water will face the aqueous water environment, whereas the water hating tails will face each other. It's a very busy place. There's lots of things happening. So it is protective in nature, but one of the biggest properties of cell membranes is that it is selectively permeable or semi-permeable. It will allow some substances to enter the cell, and it will stop some substances from entering the cell. The inside of the cell, the liquid part and the organelles, is called as the cytosol of the cell. So the cell membrane is providing structure, support, and it is monitoring what's entering the cell and what's exiting the cell. So the model for cell membranes is called the fluid mosaic model of cell membranes. So it has a phospholipid bilayer. It has some cholesterol molecules that play a huge role in the stability of the membrane. The bilipid layer is not set. It's very dynamic. It's in fluid motion. Sometimes the phospholipids can also flip-flop here. And then there is a mosaic of proteins and carbohydrates. So some proteins will go through the entire bilayer. They are called integral proteins. Some proteins will sit on the top. They are called peripheral proteins. The peripheral proteins on the inside of the cell will be attached to the fibers. They are called cytoskeleton of the cell, thereby giving it structure. These proteins can also be attached to carbohydrates or lipids, like glycolipids. And they will play a huge role in cell-to-cell adhesion and also cell communication. So what are some of the main properties of cell membranes and how molecules will move over the membrane? The first set is called passive transport, meaning it does not require energy in the form of ATP. So in simple diffusion, it refers to the movement of molecules from high concentration to low concentration. So it is just going along the concentration gradient until equilibrium is reached. So gases like oxygen and carbon dioxide can easily wiggle in and out of the cell. And also lipid-soluble molecules can move easily. When we refer to the movement of water over a selectively permeable membrane, we call it as osmosis. Why is this important? A cell can either be in an isotonic solution, where the amount of water entering the cell and leaving the cell is the same, isotonic solution, or in a hypertonic solution where it can like imbibe too much water, which is not good because it can burst, or in a hypertonic solution, which situation where it can lose water and shrivel up. So this is a picture of red blood cells, and the ideal situation for a cell to be is an isotonic solution. When a protein is involved in moving a molecule, a large molecule that can't enter the cell, like glucose, then it can use the help of a protein, a membrane protein that's found in the phospholipid bilayer. So this is called facilitated diffusion. Ions can move this way. So you can see how the protein in the cell membrane is going to bring in glucose. It's going to grab it from outside the cell and push it inside the cell. Here are some examples of active transport when we have to move substances against the concentration gradient. So this requires ATP, and our body is going to move sodium, potassium, calcium, and magnesium using energy. One of the most important that you'll study is the sodium potassium pump. So you can see this protein. It is going to, for every 3 molecules of sodium it pumps out, it brings in 2 molecules of potash. And then this requires ATP. This is called the sodium potassium pump. When we are talking about bulk transport across the membrane, see, nutrients have to come in and toxins have to leave the cell. So there are 2 main bulk transport mechanisms. Endo means bringing it inside. Cyto is cell. Endocytosis is bringing in materials inside the cell. And exocytosis would be to push out materials outside of the cell. So here you can see in phagocytosis, also called cellular eating, the cell wants to bring large particles of materials inside. So what happens is the cell membrane will like invaginate, and it will pinch off and bring it in a vesicle. So this is called cellular eating. And if it's liquid, it's called pinocytosis. If a receptor is required for endocytosis to happen, so a ligand will attach to the receptor, and then endocytosis happens, and then it's called receptor mediated endocytosis. When materials have to leave the cell, it is called exocytosis. Let's say the cell has made some hormones and it want to expel it out into the bloodstream, then that can happen through exocytosis. So this is how molecules move over the membranes in order to maintain homeostasis. So this is like a summary, a table which is very thorough, where you can like revise what is diffusion and osmosis and the different transport mechanisms. So what are the different organelles that are found in a cell? So this is a typical like a cartoon of a human cell. So it's like different types. We have different types of cells in our body, right? The cells in our brain is called neurons. So they will be modified depending upon the function, but this is a typical cell. So in general, there are 2 types of organelles. One have membrane, so they are called membranous organelles. They are isolated in the cytoplasm. They will have their own membrane. So examples of this is endoplasmic reticulum, ER for short, Golgi, lysosomes, peroxisomes, and mitochondria. So you can see that it has its own cell membrane. Nucleus has its own membrane. Non-membranous organelles do not have a membrane. So these would be the proteins that are hanging around in the cytoplasm of the cell, called cytoskeleton, microvilli, cilia, ribosomes, centrioles, and proteosomes. So here there's a cell, and then it has a cell membrane, phospholipid bilayer. The inside liquid part, jelly-like matrix of the cell is called cytoplasm, and in it are the different organelles. These networks of tubes, which is like a transport system in a cell, is called endoplasmic reticulum. The large organelle is called nucleus. Inside the nucleus, there's a nucleolus, and this is where the genetic material is protected, the DNA. And here we also see the bean-shaped mitochondria, the stack of um, structures called Golgi, lysosomes. So usually, in order to remember the cell structure and function, people can compare it to either a factory. Mitochondria is the powerhouse of the cell, which is like the generator in a factory. The nucleus is the boss. It's going to control all the activity in the cell. Endoplasmic reticulum, it's like a conveyor belt. It makes all the proteins. Golgi apparatus, it's like the shipping and handling center of the cell. So it processes the proteins, and it will put a zip code on the proteins, asking the proteins to go to a specific organelle in the cell or outside the cell, and so forth. So a good analogy is a factory in order to remember the functions of the cell. So here we have the cell membrane. The matrix is the cytoplasm. We have the nucleus, which has the DNA. Nucleolus makes a ribosomal RNA. Ribosomes are the dots that are shown here. They are the play a role in protein synthesis. Two types of endoplasmic reticulum. If it has ribosomes on it, it's called rough ER. If there is no ribosomes, it's called smooth ER. Golgi apparatus. Lysosomes are like janitors in a cell. They will clean up debris and they will recycle. They also perform programmed cell death or apoptosis. Peroxisomes, they play a role in getting rid of peroxide. It's like a spy in a cell. Mitochondria is where energy is made. The one cell in our body, the human body that can move, has a flagella, which would be the sperm cell. Cilia refers to cytoplasmic extensions. The membrane will extend to form short cilia to sweep substances over it. Microvilli are again extensions of the cell that will increase the surface area of the cell to absorb nutrients. Centrioles are found like very outside the nucleus here. They will play a huge role in making microtubules that is required for cell division during mitosis and meiosis. We'll look closely at each one of them. So here's a picture of the endoplasmic reticulum, or ER. It's a huge network or array of membranous sacs. It comes in two types. The rough has ribosomes in it, and the smooth does not have ribosomes. So here we have a clue. So because the rough ER is studded with ribosomes, it will play a huge role in protein synthesis and transport. Smooth ER plays a huge role in making phospholipids, some steroid hormones. Both of them will play a huge role in regulating the calcium concentration in the cell. It can metabolize carbohydrates, and it can also break down certain toxins. This is an electron microscope picture of the endoplasmic reticulum. The Golgi apparatus, see, it's closely situated right next to the endoplasmic reticulum. It's a stack of membranous tubes. The side that is facing the nucleus is called cis side, and then the other side is the trans side. So when proteins are made in the endoplasmic reticulum, they will pinch off in little vesicles, and these vesicles will fuse with the Golgi apparatus. And as it moves through these passageways in the Golgi apparatus, it will be processed and it will be folded to a functional form. And the Golgi is like the shipping and handling center of the cell. So it will add a sequence that will tell the protein where to go. So it's like it'll add a zip code to the protein. Oh, if it's a protein that has to go to the nucleus or a protein that has to go to the lysosome. Let's say the protein has to leave the cell, it'll pack it in a vesicle. The vesicle will fuse with a plasma membrane and then it's spilled into maybe the blood or the artery here. The lysosome that is found in a cell is will contain digestive enzymes. So it is like the janitor in a cell. It is the waste management of the cell. So because it has harsh digestive enzymes, it can fuse with other old and unnecessary structures in the cell, and it can get rid of it. So it can clean up the cell. So some of the vesicles that leave the Golgi can be enzymes that has to go to lysosome. Here is a mitochondria. It has like a bean shape. It has two membranes. The outer membrane is smooth. The inner membrane is thrown into folds called cristae. The inside part, the liquid part, is called the mitochondrial matrix. And the space between the two membranes is called intermembrane space. So the mitochondria is the powerhouse of the cell. This is where most of the ATP requirements of the cell is met. So the chemical reaction that happens here is called cellular respiration. It is aerobic. So the chemical reactions in the mitochondria can happen in the presence of oxygen. So the food that you eat is converted to ATP in three main steps. Glycolysis happens in the cytoplasm, where a 6-carbon glucose is broken down into two 3-carbon pyruvate molecules. Then there is a transition step where it becomes acetyl-CoA, and then it enters the mitochondrial matrix, where the second step, the citric acid cycle, also called TCA cycle or Krebs cycle, happens here. The third step is the electron transport chain that happens in the inner mitochondrial membrane. So glycolysis and citric acid cycle will make electron carriers called NADP and FADP. And they will enter the electron transport chain. And this is where most of the ATP will be made. So the glucose that we get through food will combine with oxygen. So oxygen is very electronegative. It's going to grab on to the hydrogen from glucose. And then ADP will be converted to ATP, which is the energy currency of the cell. Byproducts are carbon dioxide and water. So the final electron acceptor here in the electron transport chain is oxygen. So that's why it's aerobic metabolism. So if there is anything should go wrong with the mitochondria, if it malfunctions, and then it manifests as chronic fatigue because you don't have energy, ATP, required to do work. Nucleus. It's referred to like the boss of the cell. It's going to oversee all the function of the cell, and also all the anatomical and physiological activities of the cell is controlled by the nucleus. So this also has a double membrane. The matrix, the cytoplasm inside the nucleus is called nucleoplasm. And you can see the DNA in the form of chromatin. If it gets condensed, they'll call it chromosome. There is a darkly staining region called nucleolus, and this is where ribosomal RNA and ribosomes are assembled. So the picture in the bottom, it's talking about, depending on the stage of the cell cycle, you might either be able to see a X-shaped chromosome, which is highly condensed DNA. So the DNA double helix wraps itself around proteins called histone proteins. They will condense on top of each other in order to form the arms of the chromosomes. So these are called cystochromatids, and they are held together in the waist region by a protein called kinetochore. or sometimes they are found loose in the nucleoplasm. It's called chromatin. The space between the two membranes is called perinucleus space. And in the double membrane, there are these pores. They are called nuclear pores. So when a DNA want to express itself to form a protein, it will make a messenger RNA, and the messenger RNA is going to wiggle out through these pores into the cytoplasm where protein synthesis happens. Other structures are microvilli, which like you would find this in abdominal cells where absorption has to happen. Centrioles will play a huge role in cell division. They form the spindle apparatus in cell division. They make tubes, the spindle fibers. Cilia are small hair-like extensions that can. This is not cilia. This is microvilli. Cilia is a little larger than this, so they can move fluids over its surface. For example, the epithelium that lines our throat has cilia, so thereby we can move mucus over the cells. Ribosomes, they usually have two subunit, a large subunit and a small subunit. They play a role in protein synthesis. Proteosomes, they contain enzymes called proteases that can digest proteins and mainly for recycling purposes. Peroxisomes, they are membrane bound organelles and they play a huge, it is the spa of the cell, it is the detox center of the cell. So they will detox toxins and they play a role in lipid metabolism also. They are rich in an enzyme called catalase, so it's able to break down hydrogen peroxide to water and oxygen. Cytoskeleton, like we have a skeleton, and that's why we are able to keep our posture and structure. Very similarly, how does a cell keep its structure? It's because of these, proteins. They form the cytoskeleton, and depending on the thickness of the protein, they're called microfilaments is about 3 to 6 nanometers. They are composed of a protein called actin, and they provide mechanical strength. And then, along with myosin, they'll play a huge role in muscle contraction. Intermediate filaments, as the name suggests, are mid size. They are very durable. Example is collagen. They will help the cell to maintain its shape and it can stabilize all the internal organelles. It's about 10 nanometers in diameter. Microtubules are the largest ones, they are 23 nanometers. They are large hollow tubes of the tubular protein. They will play a huge role in cell division because they will be made from the centrosome. They can anchor organelles, they can change the shape of the cell, and they can also move vesicles within the cell. And they will play a huge role in the spindle apparatus during cell division. So, this is the framework of the cell. This is what gives the cell its shape and structure. And then here in fluorescent microscopy, you can see all it's lighting up in the microscope. Cell cycle. So every cell in our body is in one of the stages of cell cycle. Some cells are dividing very fast, like skin cells, whereas your neurons are hardly dividing. So the rate of cell division in different cells of the body is different. So there are two main stages in cell cycle: the interphase, which comprises of G1, S, and G2 phase, and then mitotic phase. G1 phase is when all the cell organelles will double and the cell is growing. And DNA synthesis is when DNA makes a copy of itself. And G2, it continues to grow. All the proteins required for mitosis will happen here. And then when it gets the signal to enter the mitotic phase for nuclear division, it will enter the mitotic phase. When we say cancer, so every stage in the cell cycle, G1, G2, S, they all have checkpoints. So there is a M checkpoint, G1 checkpoint, G2 checkpoint. So the regulatory molecules like cyclin, so it can move on from G2 to M only if at a certain concentration of these regulatory molecules. So it's tightly regulated, cyclins and cyclin-dependent kinases. So cancer is something going wrong with the cell cycle. So we can see that one of the checkpoints didn't work and the cell divides uncontrollably. So it forms a tumor, and then the tumor can break off and enter the blood supply, and then it can move to other parts of the body. It's called metastasis. So nuclear division, the different stages in mitosis is prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Cytokinesis refers to the division of the cytoplasm. So in mitosis, there is no change in the genetic material. So it's like taking a Xerox copy of the cell. There is absolutely no change in the genetic material. So here you can see in prophase, the chromosomes are condensing and becoming visible. This is when you see the X-shaped chromosomes. The spindle apparatus is forming and the spindles are moving to the poles of the cell. The nuclear envelope is breaking. And in prometaphase, the chromosomes are trying to line up in the center of the cell. In metaphase, they have lined up in the center of the cell and they are attached to a spindle that is radiating from the centrioles. So the nuclear membrane is completely gone. And in anaphase, they will split. So the sister chromatids will break off from each other and they will move to the poles. And in telophase, the nuclear membrane will form around each of the set of chromosomes. Cytokinesis is when they pinch off. The cytoplasm divides and the two cells pinch off. So you start with one cell and you end up with two cells which are genetically identical. So now moving on into the things that are happening in the nucleus. So here we have the DNA, which is it's a double strand, it's a double helix. And then you can see the complementary base pairing. A goes with T, G goes with C, at GC, adenine, thymine, guanine, cytosine. That is complementary base pairing of DNA. So DNA is very, very precious. It will never leave the nucleus. So when it wants to make a protein, it will make a copy of itself. It's called transcription into a molecule called messenger RNA. So one of the strands will act as a template strand, and then a messenger RNA is made. The messenger RNA then wiggles out through the nuclear pore and comes to the cytoplasm, where it will act as a template to make a protein in a process called translation. So every 3 alphabets of the messenger RNA actually relates to a protein, methionine, guanine, serine, valine. So for valine, it's GUU. That is called as a triplet code. So 3 adjacent nucleotides of the messenger RNA that translates to one amino acid is called as the triplet code. And this is universal. And that's the reason you can like cut a gene from human beings and express it in bacteria. You can like move in genetic engineering, you can cut a gene from a plant and then like put it in an animal and so forth. So this knowledge led the way for genetic engineering. So this transfer of information from DNA to messenger RNA to protein, right? This is the flow of genetic information. So when you say DNA, deoxyribonucleic acid, it is the blueprint in a cell. And this is the inheritance, the molecule of inheritance. This is what will be transferred generation after generation through the egg and the sperm. So the flow of genetic information is called as the central dogma of biology. So the process when DNA makes RNA is called transcription. When RNA is transcribed into a protein, it is called translation. Before we go into transcription and translation, it's good to remind ourselves as to how DNA makes a copy of itself by a process called DNA replication, and the method is semi-conservative, meaning one of the parental strand is conserved and a new baby strand will be made, a daughter strand will be made. So here we are duplicating the entire genome in a cell, and then like there's lots of enzymes that's involved in making this happen. So here you can see, if you unravel the chromosome, you can see the double helix of DNA. So the enzyme that is going to make a copy of DNA is called DNA polymerase. An enzyme called helicase will come and unravel the DNA. So it's going to break the hydrogen bonds between the nucleotides, and then DNA becomes single-stranded. DNA polymerase can attach nucleotides only to the 3' end. So one strand is performing complementary base pairing. So if there's a T, A will go, a G, C. So ATGC, ATGC, according to the sequence. So the leading strand is made in one stroke, and it's very fast. And because DNA polymerase can add nucleotides only to the 3' end, the other strand is made in little bits. It involves a RNA primer by primase, and then it's made in little bits. That's why it's called lagging strand. It lags in speed. And then DNA ligase is an enzyme that will come and stick up all the little pieces in the lagging strand. It's called Okazaki fragments. So this picture is showing how DNA makes a copy of itself, and the prime enzyme that makes this happen is DNA polymerase. So let's move on into transcription. So transcription refers the flow of information from DNA to messenger RNA. Let's define a gene first. So if you take any DNA, a stretch of DNA that encodes for one polypeptide, it's called as a gene. So in humans, it is speculated there's about 20 to like 24,000 genes in our body. So every gene will have a sequence called promoter sequence that's found right in front. So it's called upstream. Then the transcribing unit. So this is where the essence of the protein is. And then a termination site. So promoter, transcription unit, and terminator. So these are the three parts in a gene. In transcription, the protein that is involved, it's called RNA polymerase. So the RNA polymerase is going to glide down the DNA and looking for the promoter. And then under certain conditions are met, called transcription factors, the RNA polymerase is going to bind to it. It will gently open the DNA and then perform complementary base pairing. But there is no T in RNA. It is replaced by a base called uracil. So A will become U, T is becoming A, G is becoming C, and then it will transcribe until it hits the terminator sequence. So here's our RNA polymerase. It is looking for the promoter region. It opens up the DNA and it does complementary base pairing, and it will do so until it hits the termination sequence. So this messenger RNA, however, is not ready for being used as a template. There are expressed sequences, they are called exons, and intervening sequences. It's like unwanted DNA. So a protein complex called spliceosome will come and cut out all the introns and seal back all the exons together. So this is the messenger RNA processing. And then the environment in the cytoplasm is very harsh. So it requires a polyadenine tail, and a guanine cap for protecting it. And then it wiggles out through the nuclear pore and it comes to the cytoplasm where translation happens. So the messenger RNA will be translated to a protein. So every 3 alphabets is actually 1 amino acid in a protein. So there are 3 types of RNA. The messenger RNA, there's a transfer RNA that is going to bring the amino acid. It will have an anticodon that will match with the codon and ribosomal RNA. So what happens is the messenger RNA will lock in place. So the small ribosomal subunit will scan through the messenger RNA and it will look for the start codon called AUG, which actually translates to methionine in amino acid. So after it finds the start codon, the large subunit will come and sit on it. And depending on the triplet code found on the RNA, the tRNA, the transfer RNA that carries the specific amino acid will come and lock in place. So the transfer RNA is bringing in the correct amino acid, and then a peptide bond will be formed between the different amino acid, and the chain will grow. So there will be an exit site, a peptidyl site, and an arrival site. So in the arrival site, the transfer RNA is bringing in the amino acid, and then this will shift to the middle site. And then when arginine comes here, so it will form a peptide bond with the rest of the growing chain. So the chain will grow. So this is how the messenger RNA is translated from a nucleic acid language into a protein language. So here's a little animation that shows the codon, and then this will go on until the stop codon is hit. So how is gene expression controlled? So there are many ways of control, but transcription factors are the most important. So because every cell in our body contains all the genes, right? All the genes are found in every cell in our body, but only some genes are expressed. So there's differential gene expression. How does this happen? It's because of transcription factors. So transcription factors are proteins that will bind in the promoter region. And this binding is important for RNA polymerase to come and recognize the promoter and to initiate messenger RNA production. So this is one of the ways how gene expression is controlled. And these are the important points in the chapter, and hope that was helpful.